So organelle, structure and function. If we start with plant cells have a cellulose cell wall that's permeable but tough and strong so that it withstands the pressure exerted. If water enters that cell, it won't burst because of the cellulose cell wall. They have a vacuole to store water and other ions. They have a nucleus with a nucleolus inside. The nuclear envelope has pores to allow mRNA to leave. It has a rough endoplasmic reticulum, and that's for the synthesis of proteins. It has mitochondria because plant cells need to respire aerobically, so mitochondria allow aerobic respiration. They have infolds called cristae, which increases the surface area for aerobic respiration, and the contents is called a matrix. You have chloroplasts. Chloroplasts similarly have stacks of thylakoids intergranum, and that has a stroma on the inside. You have the Golgi apparatus, which receives lipids and it receives carbohydrates for processing as well as proteins. So it will add carbohydrates to proteins and carbohydrates to lipids. Then it will place them into vesicles called Golgi vesicles for transport to where they are needed within the cell, or for transport to the cell surface membrane for export if that's the destiny of the protein or the lipid that's come to it. The rough endoplasmic reticulum synthesizes proteins, like we said, using ribosomes. In plant cells, the ribosomes inside the cytoplasm are large, so they're 80S. And once the ribosomes have made the protein, they can be put into a rough endoplasmic reticulum vesicle, and then that can get transported to the Golgi for modification. You also have in there a smooth endoplasmic reticulum for the synthesis of lipids, so there's no ribosomes on its surface, that's why we call it smooth. Okay, if we then look at the animal cell in contrast to the plant cell, you can see that there is no wall, there is no permanent vacuole. We have a Golgi for processing and packaging, we have a mitochondrion for aerobic respiration, we have also smooth endoplasmic reticulum for synthesis of lipids, nucleolus for the synthesis of ribosomes, and we have the nucleus containing the DNA material, nuclear pores for the movement of mRNA out of the nucleus, a cell surface membrane made up of phospholipids and proteins. But in addition, in the animal cell, we also have lysosomes, which contain enzymes, hydrolytic enzymes, able to break down molecules from, for example, pathogens into smaller molecules that can be then absorbed into the cytoplasm. Also within the cytoplasm, you have centrioles, and these make the spindle fibres for making, for part of mitosis to allow separation of the sister chromatids. You have microtubules as part of the cytoskeleton that allows the movement of the membrane to allow the cell to change shape. For example, you would have a lot of those in a macrophage so that it could engulf a pathogen and hydrolyse it and digest it and break it down. So you also have vesicles, again, Golgi vesicles that can fuse with the membrane. During the exocytosis, the vesicle will fuse and release its contents, which will then diffuse out. You have endocytosis, again, that needs ATP, so that's a bulk transport, and that's the formation of a vesicle when something comes in. Phagocytosis is a specialised example of endocytosis, and exocytosis, an example of that would be in neurotransmitter release in the neurons as well. So that's cell organelle, structure and function. Again, anywhere where you see folds of anything increases the surface area, so in the mitochondria and in the chloroplast you can see lots of surface area for the reactions that they take, that they coordinate.